Good afternoon, class. Today we will be looking at homologies. We will be reading from our textbook starting on page 473. Looking at this section, I noticed words that are darkened, so they must be important for this section. As I preview the text, I also noticed that there are sections within the section in the chapter which are italicized. I also noticed that there are images above the text as well as below and which have writing as well. Okay. A second type of evidence for evolution comes from analyzing similarities among different organisms. Based on reading that first sentence, I know that homologies must be important to evolution because the first words say a second type of evidence. So that must mean that homologies is a section within evolution. I also noticed that it mentions similarities among different organisms. This reminds me of humans, which are all the same species but may have different characteristics, such as hair color. As we discussed, evolution is a process of descent with modification. Characteristics present in an ancestral organism are altered by natural selection, and its descendants over time as they face different environmental conditions. As a result, related species can have characteristics that have an underlying similarity yet function differently. Similarity resulting from common ancestry is known as homology. I still don't know what exactly homology means, but it does have the same prefix as homophones. I know that homophones are words that sound similar but mean different things. That must mean homology is dealing with similarities. In this section, an understanding of homology can be used to make testable predictions that explain observations that are otherwise puzzling. Anatomical and molecular homologies. The view of evolution as a remodeling process leads to a prediction that closely related species should share similar features, and they do. Of course, closely related species share the features used to determine their relationship, but they also share many other features. Some of these shared features make little sense except in the context of evolution. I'm confused at this part. How can features be shared and show relationships between species, but also make little sense? I'm going to move on. Forelimbs of all mammals, including humans, cats, whales, and bats, show the same arrangement of bones from the shoulder to the tips of the digits. Even though the appendages have very different functions, lifting, walking, and swimming and flying, such striking anatomical remnants would be highly unlikely if these structures had arisen anew in each species. It says that mammals share similar bones throughout the arm that may be used for different functions such as flying, swimming. I also noticed that I only talked about the subgroup mammals, which must mean that homologies are characters of a group. Looking at figure 22.15, before reading the text of the image, I noticed that bones that are similar are color-coded and look different. As I move along, I also noticed that some bones might become longer or shorter depending on the function of the limb. Okay, so looking at this, I can connect this with the human tailbone and a dog tailbone. The human tailbone is no longer used and therefore has become smaller, while the dog tailbone remains long and is used for balance. Rather, the underlying skeletons of the arms, forelegs, flippers, and wings of different mammals are homologous structures that represent variations on a structural theme that was present in their common ancestor. I know from reading the paragraph before that homologous means similar in position or structure, but not necessarily in function. I already know that a structure is like a tibia, so I know that homologous structures are just examples of what makes species homologous. Comparing early stages of development in different animal species reveals additional anatomical homologies that are not visible in adult organisms. For example, at some point in their development, all vertebrate embryos have a tail located posterior behind to the anus, as well as the structures called phalangeal throat arches. Figure 22.16. Looking at the figure, I understand that individuals can be homologous and have homologous structures, even though the structure may not exist anymore. These homologous throat arches ultimately develop into structures with very different functions, such as gills in fishes and parts of the ears and throat in humans and other mammals. Some of the most intriguing homologies concern leftover structures of marginal, if any, importance to organism. These vestigial structures are remnants of features that served a function in the organism's ancestors. I know what structures are, but I do not know what vestigial is. On Google it says a organ or a body part that has become functionless in the course of evolution. This reminds me of the appendix in humans, which has become a structure in which we do not use and can live without. Skeletons of some snakes retain vestiges of the pelvis and leg bones of walking ancestors. Another example is provided by eye remnants that are buried under scales in blind species of cave fishes. We would not expect to see these vestigial structures if snakes and blind cave fishes had origins separate from other vertebrate animals. Biologists also observe similarities among organisms at the molecular level. All forms of life use essentially the same genetic code, suggesting that all species descended from common ancestors that use this code. But molecular homologies go beyond a shared code. For example, organisms as dissimilar as humans and bacteria share genes inherited from a very distinct common ancestor. Some of these homologous genes have acquired new functions, while others, such as those coding for the ribosomal subunits used in the protein synthesis, have retained their original functions. It is also common for organisms to have genes that have lost their function, even though the homologous genes in related species may be fully functional. Like vestigial structures, it appears that such inactive pseudogenes may be present simply because a common ancestor had them. Looking at this, I can see that homologies can happen at both at an organism level and a molecular level. I can infer that a pseudogene is a gene that is not used because the text mentions that it is similar to a vestigial structure. After reading this section on homologies, I can see how homologies is a section in evolution in that it shows the relationships among organisms who are closely related. I know that homologies can be both structural as well as molecular. I wonder if there's a way in which homologies are used when creating relationship charts.